Dr. Jones checking in. This is A&P 1 Lab Test 1, and we need to cover the phases of mitosis as they show up on the microscope slides. So here we are. Let's see, let's make sure the pen is turned on. It was not. Okay. So here we have a cell that's in interphase. And yes, I'm writing with a mouse. Sorry. I couldn't get the pen or any of that other stuff to work. So we're writing with a mouse. Interphase. So interphase is the part of the cell's life cycle when it is not undergoing mitosis, so it's not dividing. So this is a non-dividing cell. You've got a clear cell membrane, you've got a nuclear envelope, and you can see right there the nuclear envelope where I haven't drawn it in. And no visible chromosomes. There are some inclusions, but nothing that we're concerned about. And next is the first phase of mitosis is prophase. In prophase, the nuclear envelope dissolves, the chromosomes coil and then supercoil into visible chromosomes. So the DNA in a non-dividing cell is in chromatin. It's that red fuzzy stuff we saw in the nucleus on the cell model. And it's not visible in a light microscope because it's too thin to show up. The chromosomes, when they've coiled and then supercoiled, and when I say they've coiled, it's like if you've ever twisted your shoelace. Like just keep twisting, and then it makes like a spiral that's coiled. So then the coils coil, and that's the supercoil. And then that's visible chromosomes, and that's what all these squiggles in here are. These are visible chromosomes just hanging out in the nucleus of the cell. Same thing here and here. So these are all prophase. Trying to show you, here we go. And next we have metaphase. Trying to give them in order the first time. Metaphase. And in metaphase, the chromosomes are lined up in the middle on the metaphase plate. So these dark structures are chromosomes. The line of them is the metaphase plate. And there will be centrioles at either end, and they'll be attached by spindle fibers, which we can sort of see. You sort of see the little spindle fibers. Okay. And then here's our next phase of mitosis, which is anaphase. Metaphase lined up in the middle, anaphase pulled apart. So, apart, poop. Okay, and then there's an eraser, eraser. Boom, boom, boom. There it is. And then back to the pen. Okay, anaphase, they get pulled apart. Okay, so what happens here is the metaphase, the chromosomes are lined up in the middle of the metaphase plate, and then in anaphase, the spindle fibers that attach them to the centrioles get shorter, they shorten, and so they pull the chromosomes apart. And so here you see they kind of look like teeth because they're being pulled apart. Okay, so there's anaphase. Okay, and then here's another anaphase. What else do we? Here's another anaphase. Okay, so our phases of the cell life cycle. Interphase is when the cell is not dividing and it is not part of mitosis, but it is part of the cell life cycle, so I'm going to include it. And then next is prophase, metaphase, anaphase, And the last will be telophase. So our mnemonic here is I pass my anatomy test. So let's see if we can find a telophase. Here we are. Okay, so in telophase, in late anaphase, you start a process called cytokinesis. And the process is indicated by this divot. So the divot is called a cleavage furrow. Cleavage is cell division. Cleavage furrow. But the process indicated by the cleavage furrow is cytokinesis. There should already be a video where I go through a lab video where I lecture on the phases of mitosis. So here we'll just cover the slides. The cytokinesis, mitosis is the division of the genetic material between two daughter cells. Cytokinesis is the division of the cytoplasm between two daughter cells. Cytokinesis does start during anaphase, but clearly hasn't started here because there's no cleavage furrow. So this, you could call this early anaphase, but that's unnecessary. Anaphase is fine. And then we will call this telophase. And in telophase, the cytokinesis continues. The cleave, forgot the edge, A, G, E, cleave edge. Furrow, okay. And the chromosomes will uncoil back into chromatin, and you do start to reform the nuclear envelopes. Let's see what else we have. Okay, and here's another. What's the indent called? A cleavage furrow. What process does that indicate? Cytokinesis. So we can point right here and ask two different questions. So please, please, please double check that you have answered the question that was asked. Because if it asks for the process that this indicates and you say cleavage furrow, that is wrong, because it does not correctly answer the question. If it asks for what is this indent and you say cytokinesis, that's wrong, because the indent is the cleavage furrow. The cytokinesis is the process that that cleavage furrow indicates. Ooh, what was this one? Metaphase. That's a name. Okay, we will come back to that in the next video. We will cover epithelial cells in their own video because slides. Yeah, it's a challenging bit, so we'll cover it in smaller chunks. Okay, so this has been A&P 1 lab test 1, the phases of mitosis microscope slides video. There's gotta be a shorter way to say that. In any case, it's good to see you here. Thank you again for tuning in. The education must go on. Dr. J out.